to introduce our eminent speaker of this evening, Dr. Mangesh Tiwaskar sir, who is the gold medalist, who is also Honorary General Secretary of National API, ICP, and PRF. He is on editorial board of JAPI Medicine Update. He is the guest editor in various journals, delivered more than 1,000 lectures at the many national and international conferences, and has a multiple chapters, publications, posters, presentation, write-up in the next books. Very warm welcome to the Mangesh Tiwaskar sir to this beautiful session. Over to you, sir.
Thank you very much, Dr. Dhananjay. And Purvi and Manoj, I'm really jealous of you guys. Such a wonderful and such a superb program that you have continued. And I'm really failing short of words to appreciate last panel discussion. Sunil, Unni, and Anuj, along with Purvi, did a brilliant job. There are so many questions that I also had, and you technically answered everything. So moving on, as today being the special day, and as Manoj has already talked about, we are celebrating the centenary year. Now, as we know, we have got original insulins to manage our diabetic patients correctly. But we all know that, the innovator insulins or any innovator biologics, if at all we talk about, has some caveats as well. And most of the caveats are usually the availability, cost of the therapy, and maybe possibly, the I would say outreach to the deserving population. And hence, probably the concept of biosimilars have come. So in next 15 minutes time or so, as Manoj has asked me, I'll try and cover what are the basics of biosimilars and what are the things that you're supposed to do when you talk about interchangeability. Are there any laws and what we are supposed to know as far as the biosimilars are concerned? I don't have any disclosures. And in my agenda for today, after a few introductory remarks, I'll just give you biologicals, what are biologicals and why you are supposed to know about them. I'll just give you a bird's eye on that. And then probably open mind thought to biosimilars. Let us have an open mind discussion as far as the biosimilars are concerned, and few carry home messages or carry about messages that I'll give. So remember that when any biological or any drug is invented, there are two ways of production of the drugs. Number one are what we call it as a small molecules, or also called as generics, are usually chemical compounds. And the other group are the large molecules, usually called as biologicals, and they are actually derived from the living cells or proteins. Now, there are so many sources of biologics. I'm not going to go to the details about it, but remember that, and especially in the COVID times, we already have realized the importance of biologics. So this is the need of the hour today, and the world is going to need them. And remember that these biologics are the large molecules. They are not only just large molecules, but they are far more complex than the simple chemical structures and chemical pharmaceutical agents that we are supposed to use or we are trained to use. More importantly, they are the need for the treatment of the most difficult conditions or serious conditions like malignancies or autoimmune disorders or some neurological disorders or some hematological challenges. Biologics, and including COVID now, biologics have changed the way we actually look at the pharmacotherapy of these difficult medical conditions. And most importantly, biologics are typically made in the living organisms mainly by what we call it as the genetic engineering or a DNA technology. So these are the typical medicines which are usually grown up from the living cell line. And a cell line is expanded, the cell line is picked up, the cell line is isolated, the DNA is introduced into the cell, and this cell line is expanded into bioreactors and then ultimately gets purified and is available to the world for use. Understand this very intriguing fact that typically from the birth of the idea to the prescription table, it will take around nearly 17 to 15 years for a drug to reach the patient. So from the invention of the concept or the thought of the concept to the inception of the drug, this much period is required. And look at the cost involved, around 150 to 300 million dollars. Very interesting fact, I'll bring it to your notice. If it will not be an exaggeration if I say that it is more difficult and expensive to reach human body than to even reach the outer planet. Remember Mission Mars? It took just 3 years and $70 million. So that is the importance of, I would say, pharmacological research. Now, why the biologics are difficult to produce? Now, you can see on the left side of the screen, Look at the small generic chemical molecule of aspirin, which is just 180 Daltons. This is a very typical chemical drug that you are able to see on the screen. But when it comes to a biological, large, nearly around 150,000 Dalton molecule, which has got more than 800 folds, are so difficult to produce. And hence, you need to understand the differences. So overall, biologics per se are classified into two larger groups. Apart from the small chemical molecules that we know about, are usually the intermediate molecules and the large molecules. Intermediate molecules has insulin. They are mostly hormones or growth factors. They are complex and difficult to replicate, but it is not impossible to replicate them. But monoclonal antibodies, which are large, highly complex molecules, are technically impossible to replicate, and you cannot make the biosimilars of these monoclonal antibodies. All the biologics are difficult to copy, but it is not impossible. And remember that there is a method by which the entire copying happens. The challenges for the innovator biologics are usually they have to undergo very tedious DCGI FDA clearance norms. And that is the reason why you have seen that, the concept takes nearly 15 years of investment of nearly $300 million. So they become expensive and not all can afford them. And also requires a very highly sophisticated production and technology, and mostly technology is patented. And that is the reason why biologics need some biosimilars, and especially intermediate biologics. They will definitely, be needed as a biosimilars to make them more affordable. And as the patent expires, really it also offers a better competition between the various pharmacological industries. So what are biosimilars? They are the biological medicinal products which will contain the same version of an active substance of an already authorized original biological medicinal product, that we always call it as a reference medical product. And this biosimilar word primarily was born from European Medicine Agency, or EMA, as a regulatory term. There are a few other terms, like WHO use it as SBP, that is similar biologic product. US FDA uses as follow-on biologic. Canada uses at SEB, that is subsequent entry biologics. So there are different definitions of these biosimilars. And look at this. The concept was introduced to the world in 2004 from Europe. And in India, it was introduced in 2012. And from that time onwards, India has become one of the global hub for the production of the biosimilars. And India is one of the leading contributors in the biosimilar markets, and more than 50 biopharmaceutical brands are marketed whole over India, and it costs around $220 million. And this is what we know about biosimilars, right? But remember that, and this is a very important slide that I want you people to understand, that biosimilars are not generics. They are different. They are highly complex. The complexity is significant as far as the size, the post-transcriptional modifications, the flood patterns, the antigenic drift, everything occurs to the biosimilar. So they are different from generics. They cannot be classified as generics. So biosimilar in nutshell are different from the host cell series of the innovators. Remember that. But most importantly, manufacturing expertise is needed. And even though it is different, but still, even the cell line is different from the innovators, but still the expertise is needed. There can be a batch to batch variability, but that can be even with the innovator biologics. And understandably, innovators always have a tendency to keep their recipes in a lock and key, secret recipes of the innovators, naturally. And hence, sometimes biosimilars can have immunogenicity. And there are few causes for the immunogenicity. And that is the reason why biosimilars need to prove their safety. And hence, a robust pharmacovigilance is needed when you talk about biosimilars. Now, why we need biosimilars? I've already discussed that these are affordable molecules. They bring down the cost, and the approval process is much faster. Now look at this. As the patents expire, naturally the competition for getting the biosimilar comes into. And remember that the production process, even though it is almost similar, but the post-production approvals are much less expensive and much more quicker. Now why this happens? Because you can see that a reference biologic or an innovator biologic, there is a huge methodology of developing the product, right from the molecular characterization to the preclinical trial to phase 1, phase 2, phase 3, and then the post-marketing surveillance happens. All that can be bypassed when you are manufacturing a biosimilar. And what you are supposed to do are to have a few around, say, phase 3 clinical trials or a phase 4 clinical trials, but you need to have a robust technique of developing a molecular characterization, And then the post-marketing survey, or what we call it as a pharmacological vigilance or a pharma vigilance, is mandatory. Always remember that all biosimilars need to satisfy the extensive analytical characterization that is usually a character of the production of any biologic, no matter it could be an innovator biologic or a biosimilar biologic. And that is the reason why, being in India, we have a huge human resource to come up with the biosimilars that we have, and especially insulins. Now, this is what the cost-effective production of biosimilar is, that you do not require any animal studies, you do not require any phase 2, phase 3, phase 1, phase 2 studies. Phase 3 studies are only really needed to just prove the non-inferiority of the product to the innovator biologics. Preclinical studies are not necessary and they are not so robust as innovators. Approval process is relatively simpler, and most importantly, they do not require too many clearances, and that offers a better competition. And this is what the paper which was published by Dr. Sanjay Kalra and team from the Sears group of the people, and they have clearly told us what biosimilars should satisfy and what is the switch of biosimilars. Interestingly, a very simple and a beautiful thing which has come up in a quest to develop biosimilar in one of the monoclonal antibodies, that when they were working on with infliximab, which is a monoclonal antibody used to treat the rheumatoid diseases or an arthritis type of a diseases or autoimmune diseases, they actually created a better product which is much better, and these are called as biosuperiors or biobetters. Now remember that. Or a filgrastim was worked, and then during the working of biosimilar of filgrastim, they ended up finding out pegfilgrastim. Remember that if there is a biobetter production, all the similar laws and the approval processes are needed similar to the innovator biologics. There are few biomimics where in the countries where you do not have a very stringent approval mechanisms, you get something called as biomimics or biocopies. They are not as stronger biosimilars as, because you can see that high quality biosimilars need to satisfy nearly 95 similar attributes, where a low quality biosimilar, or what we call it as biological copies or biomimics, they do not require so much of attributes and they require just 87 attributes in the acceptable things. In the last 2 or 3 minutes, I'll talk about biosimilar insulins, especially in the Indian context, and the Biocon insulin that we have, because Biocon being the educational partner for this session, I'll talk about their insulin. As the manufacturing is complex with the biologics, same way insulin manufacturing is also not easy, and it's a very complex procedure. And remember that the cell lines which are used for the production are usually either yeasts or E. coli bacteria, which are usually processed, and then you get a biosimilar or an innovator biologic. Now, whenever you talk about the biosimilar, the question of immunogenicity and effectivity is always asked. And that is the reason why this very interesting poster, which was published in IDF in 2015, brought or caught my attention. And you can see that in comparison to the innovator Lantus insulin, look at the immunogenicity of Glaritus as well as Basaloc. You can clearly see that Lantus and Basaloc technically go hand in hand as far as an immunogenicity is concerned. And Biocon's insulin has also conducted the global phase 3 trials on various groups of the diabetes patients, and also they have conducted the trial which is called as InStride 1, 2 and 3, where they are not only looked at the effectivity of this biosimilar glargine of Biocon in the type 1 diabetic patients, type 2 diabetic patients, and they also have looked at some patients where the switch was done from the innovator biologic insulin to the biosimilar insulin of Biocon. And what they found out, that as far as the glycemic control is concerned, it was almost no difference observed. As actual over the period dose required was concerned, it was not different than the innovator insulin. All time or nocturnal hypoglycemia was concerned, it was almost similar to the original insulin. And most importantly, when they did a switch from the innovator insulin to the biosimilar, they found out that there was not too much of immunogenicity challenge. The safety, the efficacy, the characterization remained almost similar to an innovator insulin. Lastly, talking about substitution, are there any laws? Yes, remember that we being in India practicing, there are very clear laws of what we call it as an interchangeability or switch. always instruct your pharmacist that they should not switch or interchange the insulin brands or any intermediate biologics brand to the biosimilars if at all they are available. There has to be a clear instruction to the pharmacologist or a pharmacist on that. And remember that the prescriber should be able to prevent the substitution. So it is your job to prevent the substitution by writing down dispense as written or brand medically necessary. Prescriber should be notified by the pharmacist about the switch if at all it is happening, and simultaneously it is your job and the pharmacist's job to inform patients also that they are doing a switch for whatever the best possible understood reasons. So friends, to summarize, I would say biologics are the need of the hour. The process is complex and requires a huge amount of investment, time, and expertise. Biosimilars have, especially when it comes to the intermediate molecular biologics, I can tell you that biosimilars have offered a more affordable choice and giving a drug which is almost identical, not even though similar, I would not say, but nearly identical to the innovator biologics. Biosimilar approvals, they are easier, they are cheaper, and hence biosimilars, they become much cost effective options for the therapies, especially in those class of patients where we have a huge load of diabetic patients, and we being in India, we are all low socioeconomic status where our authorities, our governments do not invest too much in the healthcare sector probably. And when the healthcare sector is actually dealt by the patients and they pay from the pocket, it becomes imperative to address the cost as a challenge. And hence, I appreciate Bansi and the team when they have for the first time appreciate the cost factor and included that in the RSSDI diabetes management guidelines, where they said that cost is one of the biggest thing that has to be considered when you are offering any kind of the glycemic lowering therapies or any medical therapies to the patients. And hence, physicians, patients should have a choice, and their decision has to be critically taken when they want to prescribe biosimilars and while making the use of biosimilars in the process. Thank you very much. I wish you all a very, very happy and a prosperous new year and a very happy festive season. Purvi, Manoj, you guys have rocked today, and I'm very happy to see Dr. Beni Madam as well. Good evening, Madam.
Thank you. Good evening. Wonderful talk.
Mangesh, thank you. I think great talk, clearing up any kind of confusion, if at all, on the biosimilars. There have been questions coming, and I'm sure the chairpersons are also checking on the link for the questions from delegates. There is one question, not directly linked to this necessary, but I thought Mangesh, you would be the right person to answer. Somebody is asking a question on quality of insulins, and can insulin be arrhythmogenic?
Arrhythmogenic. Insulin can be only arrhythmogenic when probably there are two ways by which it can trigger hypoglycemia, and that subtle hypoglycemia has presented as an arrhythmogenic trigger to the patient, especially to the vulnerable class of the patient. So you may not say that actually the insulin as a molecule is arrhythmogenic. And always remember that we naturally produced insulin in the beta cells. So how a naturally produced product can be arrhythmogenic, if at all, we need to understand. But the only arrhythmogenicity can be because of the possible hypoglycemia. And if suppose somebody has insulin sensitivity, these are the only two patients, class of the patients where you should be a little more careful.